Welcome to WikiPodcast, where we take a deep dive into the stories that shape our world. Today, as part of our History Makers series, and we're zoning in on scientific geniuses, we're uncovering a really fascinating life, one often, well, overshadowed in the history books. A true scientific genius whose work was absolutely central to one of the biggest discoveries of the 20th century. We are talking about Rosalind Franklin, the brilliant woman behind the DNA helix. It's quite a journey, filled with groundbreaking science, some surprising twists, and a legacy that still sparks discussion today.
Yeah, and what's truly illuminating here is how her meticulous experimental work didn't just contribute to understanding life itself. It really defined the foundational method that made it possible, even if, her role wasn't fully acknowledged back then in her lifetime.
Okay, right, let's unpack this. Our mission today is to explore Rosalind Franklin's remarkable journey, from her early days through her pivotal contributions to DNA and also virus research, actually. And then finally, her lasting impact. Get ready for some surprising insights, I think. So Rosalind Elsie Franklin, born in Notting Hill, London, 1920, came from an affluent, influential British Jewish family. And apparently, right from the start, she showed these exceptional abilities, doing arithmetic for fun at just 6 years old.
Wow, 6.
Yeah, right. She went to St. Paul's Girls' School, which was pretty notable because it was one of the few schools then that actually taught physics and chemistry to girls. And she just excelled. Not just science, but Latin, sports, became fluent in French, too.
Quite the polymath even then.
Totally. And here's a quirky little fact. The composer Gustav Holst, he is her music director and apparently once questioned her hearing.
Huh. Well, clearly she heard the call of science loud and clear.
Definitely. And her family's values, they seem really remarkable, too. Her father, for instance, asked her to think about giving her university scholarship to a deserving refugee student. It really highlights how involved they were in helping Jewish refugees fleeing the Nazis. They even took in children from the Kindertransport evacuations.
That says a lot about the environment she grew up in, that sense of social responsibility.
Yeah. So after St. Paul's, she goes to Newnham College, Cambridge, in 38, studies chemistry, graduates in 41. But her first shot at PhD research, under Ronald Norrish, who later won a Nobel Prize, mind you, it didn't go well. She found him difficult. I think the quote is she despised him completely.
Oof. Well, that initial friction, sometimes that pushes you in unexpected directions, right? Maybe it inadvertently steered her towards something else.
Could be, because here's where it gets really interesting. She joins BCURA, the British Coal Utilization Research Association, in 1942, part of national service during World War II.
Right, so very practical wartime work.
Exactly. Researching coal porosity, using helium to measure density, super meticulous stuff. And it led to key discoveries about classifying coal, how it performs as fuel, and, importantly, for wartime gas masks.
Ah, direct application.
Totally. And this work actually earned her a PhD from Cambridge in 1945. Plus, she was also an air raid warden during the war. Just incredible.
So she's doing this really intense practical research under wartime pressure. How do you think that focus on, real-world problems and precise measurement shaped her later work?
That's a great point. It seems like this foundational practical work did more than just get her a PhD. It must have honed that precision, especially with X-ray techniques.
Right. It cemented this scientific philosophy, rigorous data-driven investigation above all else. You can see how dealing with tangible materials under pressure prepared her for tackling something as complex as DNA.
Yeah, that background seems crucial. So the war ends, and Franklin, always looking for the next challenge, finds this new opportunity in Paris. Through a contact, she joins Jacques Méring's lab in 1947, postdoc position.
And Mering was an X-ray crystallographer too, but with a twist.
Exactly. He applied X-ray diffraction to amorphous substances, like rayon, things without that neat crystal structure. So he taught her the practical skills for that specific, complex technique, how to interpret results from messy, non-crystalline stuff. A real niche.
Okay, so she's not just learning X-ray crystallography, she's mastering a particularly tricky branch of it.
Precisely. She used these skills on coal, other carbon materials, looking at how atoms rearrange when they turn into graphite. She even coined the terms graphitizing and non-graphitizing carbon.
So she's building this unique expertise.
Yes, and this expertise, developed in Méring's lab, was about to become incredibly important. Then, in 1951, she moves to King's College London. Now, she was actually appointed to study proteins and lipids, but kind of unexpectedly got redirected to work on DNA fibers.
A bit of a sudden change in direction then.
Yeah, and she was, at that point, the only experienced experimental diffraction researcher there. They assigned Maurice Wilkins's PhD student, Raymond Gosling, to assist her. But here's a key issue. The director, John Randall, apparently didn't communicate her new role clearly, especially not to Wilkins. And this lack of clarity seems to have created friction between Franklin and Wilkins right from the get-go.
The classic communication breakdown, setting the stage for conflict. Not ideal.
Not at all. But despite that tension, her work, immediately groundbreaking.
Yay.
She refined the X-ray equipment and, crucially, she innovated this humidity-controlled camera chamber.
Why was controlling humidity so important?
Because it allowed her to discover that DNA exists in two forms. There was the wet B form, long and thin, and the crystalline A form, short and fat.
Two distinct structures from the same molecule.
Exactly. And the B form was the one likely active in cells, while the A form gave clearer diffraction patterns. Both were vital clues. Her analysis quickly showed, quote, evidence for spiral structure, and critically, that the phosphate groups were on the outside the DNA chain. That was huge.
A major structural insight.
Definitely. She presented these findings in a lecture in November 1951. James Watson was there, but apparently he didn't fully grasp the significance of her data at the time.
And what about her approach? You mentioned her meticulousness earlier.
Yes, that's really revealing here. Franklin was very much against jumping to conclusions, against premature model building. She famously believed, we're not going to speculate, we're going to wait. Let the spots on this photograph tell us what the structure is.
Right, pure empiricism, let the data lead, which is admirable science, of course.
Absolutely. But that scientific caution, maybe it meant she moved a bit slower than others, like Watson and Crick, who were perhaps more willing to speculate and build models based on less complete data.
It does raise that classic question in science, doesn't it? The tension between rigorous step-by-step proof and the sometimes necessary leaps of intuition or synthesis needed for a breakthrough, especially in a race.
That's a perfect way to put it, because it was becoming a race. Over at Cambridge, Watson and Crick were working hard on their DNA model. Their first attempt, a triple helix, totally flawed. And Franklin's data, like the phosphates being on the outside, was key in showing them it was wrong.
So her work was already influencing theirs, even indirectly.
Yes. Then comes January 1953. Watson sees the photo, photo 51, that incredibly clear X-ray image of the B-form DNA, taken by Gosling under Franklin's guidance.
And Wilkins showed it to him.
Yes, apparently without Franklin's knowledge or permission, which understandably became a major point of ethical debate later on.
I can imagine.
Watson also got access to a Medical Research Council report. It contained Franklin's detailed crystallographic calculations from December 52, things like the 34 angstrom repeat distance and the C2 symmetry.
Okay, what do those measurements actually mean for someone trying to build a model?
They're basically the architectural blueprints, the 34 angstroms. That's the precise length of one full turn of the helix.
Got it.
And the C2 symmetry. That strongly suggested two identical strands running in opposite directions.
So the exact dimensions and the arrangement needed for a double helix, critical scaffolding.
Absolutely. Foundational data they needed. Now, interestingly, by February 53, Franklin herself, working independently with her own data, was concluding that both A and B forms likely had two helices.
So she was getting very close to the final picture herself.
Very close, though she hadn't quite worked out the specific complementary base pairing, A with T, G with C, that was Watson and Crick's big conceptual breakthrough.
Right, the final piece of the puzzle.
Yeah. And actually, later evidence, like an unpublished Time article from 53 that consulted Franklin, suggests maybe it was a bit more collaborative than the stolen data narrative implies. Perhaps more of a sharing of information among the key players. She was seen as an equal player. It's complex.
History is rarely simple, is it?
Never. So Watson and Crick finished their double helix model on March 7th, 1953. They published in Nature on April 25th. Their paper just has this brief footnote acknowledging a general knowledge of Franklin and Wilkins' unpublished contribution.
Quite understated.
Very. And Franklin and Gosling's detailed paper, the one with photo 51, was published right after theirs in the same issue. So it kind of looked like it was just supporting evidence for the Watson-Crick model rather than the independent foundational work it was.
That placement definitely shaped the initial perception.
For sure. So this really shaped the narrative for a long time that her vital contributions were minimized. And that leads into all the posthumous controversy and eventually the efforts for her rightful recognition. But even as the DNA story is unfolding, Franklin was already moving on. Mid-March 53, she goes to Birkbeck College. She described it colorfully as moving from a palace to the slums, but pleasanter all the same.
So a change of scenery, maybe a better working atmosphere.
Seems like it. She worked under John Desmond Burnell, a crystallographer known for actually promoting women scientists. And there, she started pioneering work on the structure of viruses.
Right, tobacco mosaic virus, TMV, and polio.
Exactly. She shifted her focus, but brought that same rigor. This wasn't a step down at all, was it? Solving virus structures back then was a massive challenge.
Absolutely. Her skills were perfectly suited. She wasn't just looking at them, she was figuring out the actual architecture of infection at a molecular level. Really groundbreaking stuff for virology.
And she excelled. She formed this long, successful collaboration with Aaron Klug,
Who later won a Nobel Prize for continuing that work that Franklin essentially introduced him to.
That's right. Her team published seminal papers, showed TMV protein molecules arranged in the Heelys, the RNA wound inside the hollow virus. Just fundamental discoveries.
Incredible productivity and insight.
But then, tragically, mid-1956, she's diagnosed with ovarian cancer.
Oh, no.
Yet, despite being ill, she just kept working. Published 7 papers in 1956, 6 more in 1957.
Wow. That resilience. While battling cancer, it's just astounding. Shows incredible dedication.
It really does. She passed away April 16, 1958, only 37 years old. The cancer complications and X-ray exposure is often cited as a possible contributing factor.
A tragically short life, given everything she achieved and could have still achieved.
Absolutely, and a poignant detail. She'd been building this 5-foot model of the tobacco mosaic virus for the Brussels World's Fair in '58. It opened to the public the day after she died.
Oh, gosh.
So her untimely death meant she wasn't around when the Nobel for DNA was awarded in '62 to Watson, Crick, and Wilkins.
And Nobel rules back then prohibited posthumous awards.
Right, and the narrative around her was heavily influenced by James Watson's 1968 memoir, The Double Helix. It painted a pretty critical, sometimes dismissive portrait, used that nickname Rosie, which she apparently dislikes.
Yeah, that book definitely fueled the wronged heroine story for decades.
It did. But as we touched on, more recent historical work, looking at various sources, suggests a more complex picture. Less outright theft, perhaps more complex scientific exchange, with Franklin recognized as an equal by many contemporaries, even if not by everyone or in every publication.
It really forces us to think about how scientific history gets written and rewritten. It's rarely just one person's heroic discovery, but often this messy, collaborative process.
Exactly. And the Nobel situation highlights that too. Many argue it's highly likely she would have shared the 1982 Nobel with Aaron Klug for their virus work if she'd lived.
Very plausible, given the impact of that research she initiated.
Thankfully, today, her contributions are widely recognized. There are buildings, awards, plaques named after her, an asteroid, a statue, a Google Doodle, that UK 50p coin with photo 51 on it.
And even the ExoMars rover, right? Named Rosalind Franklin.
That's right. A fitting tribute traveling to another world. Rosalind Franklin's story, it's such a powerful reminder, isn't it? That scientific progress isn't always simple or linear. That the tireless, meticulous work, often done behind the scenes, is absolutely indispensable.
Definitely. She was just such a rigorous scientist, a sharp intellect, a woman who genuinely pushed the boundaries of how we understand life itself.
She really didn't just contribute to the DNA discovery. She laid down the experimental framework that made it possible. A foundation built on precision, hard data, and letting the facts speak for themselves. Thanks for listening. Write us your opinion in the comments on how Rosalind Franklin's story impacts your understanding of scientific collaboration and recognition. Subscribe to the channel to help more listeners reach our deep dives. Thank you, and have an amazing day.